Welcome to the deep dive. Today, we're going to be diving into the world of DNA organization.
Oh, cool.
You've given us some great research on viral, bacterial, and even eukaryotic DNA,
Yeah,
and it looks fascinating.
it is. It really is. It's remarkable. It's like trying to fit an entire library into a tiny box,
Right.
but on a molecular level.
Okay, I love that analogy. So
Yeah.
let's start with the basics: viruses and bacteria.
Okay.
Their chromosomes seem much simpler than ours. Right?
Yeah, exactly. They're like the starter pack of DNA organization.
Okay.
Take viruses, for example. They're incredibly diverse, but always streamlined. They can use DNA or RNA, have it single or double-stranded,
Wow.
and even choose between circular and linear structures.
So it's almost like they've optimized for efficiency above all else.
You could say that.
That's
Yeah.
okay.
Think of a bacteriophage virus injecting its linear DNA into a bacteria.
Right.
It then cleverly forms a ring,
Oh, why?
ensuring it doesn't degrade in the harsh cellular environment.
Oh, that's clever.
It's incredibly clever,
Wow, it's like a tiny, efficient machine.
right?
And then we have bacteria like E. coli with its circular chromosome.
Which is about 1.2 millimeters long,
Right,
which is a seeming contradiction considering it fits inside a microscopic cell.
right.
But to put that into perspective, if you stretched out all the DNA in a single human cell, it would be nearly 2 liters long.
Whoa. That's some serious packing going on.
Yeah.
So how do they do it? I've read about this supercoiling thing.
Supercoiling is key. It's like twisting a rubber band upon itself to make it more compact.
Oh,
Researchers discovered this by spinning DNA at high speeds.
okay.
They found tightly wound supercoiled DNA moves faster,
Wow.
almost like a tightly wound spring uncoiling rapidly.
So that's how they fit so much DNA into such a tiny space.
Yeah.
But I'm imagining tiny hands in there twisting it up.
Right.
Is that how it actually works?
Not quite. We have to thank amazing enzymes called topoisomerases for that. They're like molecular scissors, cutting, twisting, and resealing DNA strands to manage supercoiling.
Oh, cool.
And what's fascinating is this isn't just about space saving.
Oh, there's more to it.
Absolutely. You see, supercoiling actually regulates which genes are readily accessible.
Okay.
Imagine a scroll. The tightly wound parts are harder to read.
Right.
Similarly to poisomerases, by changing the supercoiling, can expose or hide specific genes controlling their activity.
So it's not just storage.
Yeah.
It's like an access control system for the DNA.
Exactly. And this becomes even more critical in harsh environments.
Oh, okay.
By tightly regulating access bacteria like E. coli can rapidly activate genes needed for survival when, say, temperatures rise or nutrients become scarce, giving them a real edge.
Wow, that's a whole new layer of complexity I hadn't considered. Okay, so we've got these supercoiling masters in bacteria,
Yeah,
but what about us eukaryotes?
right.
Our cells have a lot more DNA to deal with.
You're right, and that's where things get even more fascinating. Eukaryotic DNA organization is like going from a simple bicycle to a high-performance race car.
Ooh, okay.
Yeah.
From what I've read, it involves this thing called chromatin, right? It's kind of a mystery to me.
Chromatin is the key. It's a complex of DNA and proteins, mainly histones,
Cool.
that act like spools, winding the DNA into tidy packages called nucleosomes.
Okay.
Think of it like beads on a string. Each bead is a nucleosome with DNA wrapped around a core of histone proteins.
Okay, so that's step one. The DNA is neatly wrapped.
Right.
What happens next?
Well, it doesn't stop there. These nucleosomes coil up further into a 30-nanometer fiber, and then these fibers form loops.
Okay.
Ultimately, they condense into the iconic X-shaped chromosomes we see during cell division.
It's like origami on a microscopic scale. But with all this packing, how does the cell actually access the information in the DNA?
Right.
It needs to make proteins copy itself.
Yeah, that's a great question.
Yeah.
And it brings us to one of the most dynamic aspects of DNA organization, chromatin remodeling. You see, this packaging isn't static. It's constantly changing to accommodate the needs of the cell.
So it's like rearranging furniture in a room to get to something you need.
Precisely. Imagine a library with bookshelves that can rearrange themselves so you can always find the book you need.
Right.
Yeah.
Okay, I'm getting a clearer picture now.
Good.
But how does this remodeling actually happen?
Well, it turns out those histone proteins aren't just passive spools.
Okay.
They have these tails that stick out, and they can be modified with chemical tags.
Huh.
These tags act like switches, controlling how tightly the DNA is packed and influencing which genes are expressed.
Wait, so these tiny chemical tags can actually control which genes are turned on and off.
Exactly. It's like having a complex control panel for gene regulation.
This is all so mind-blowing. I'm starting to realize that DNA organization is so much more than just stuffing DNA into a tiny space.
Right.
It's about creating a dynamic and responsive system
Yeah.
that controls access to genetic information,
Right.
making sure the right genes are expressed at the right time.
You got it. And we're only just beginning to unravel all the intricacies of this system.
Wow.
But before we get ahead of ourselves, let's take a closer look at some of the specific players in this chromatin remodeling game.
So we've got this incredible system of chromatin remodeling going on. It's like a constant dance with the DNA packing and unpacking
Right.
to allow access to different genes.
Yes.
But I'm still a bit fuzzy on the details of how this remodeling actually happens.
It really is a fascinating dance, and the key choreographers are those histone tails we talked about.
Okay.
Remember, they can be modified with chemical tags that act like switches.
Mm-hmm.
One of the most important modifications is acetylation.
Acetylation. That sounds familiar, but can you remind me what that is?
Sure. Think of it like this. Histones are naturally positively charged, which helps them bind tightly to the negatively charged DNA.
Right.
Acetylation adds an acetyl group to the histone tail, neutralizing some of that positive charge.
So it's like loosening a belt to make it more comfortable.
Exactly.
The histones relax their grip on the DNA a bit.
Yeah, and this loosening allows other proteins to access the DNA for things like transcription,
Yeah.
the process of making RNA copies of genes.
So in a way, acetylation is like a signal that says, hey, this section of DNA is open for businesses.
That's a great way to put it. And what's really amazing is that there are specific enzymes
Wow.
that add and remove these acetyl groups.
Okay.
It's a highly regulated process, ensuring that the right genes are accessible at the right time.
This is incredibly complex. Are there other types of modifications besides acetylation that play a role in chromatin remodeling?
Oh, absolutely. Acetylation is just one piece of the puzzle.
Well,
There's also methylation, phosphorylation, and a whole host of other modifications,
right.
each with its own specific effect on chromatin structure.
So
It's like a complex language of chemical tags,
okay.
fine-tuning the accessibility of DNA and ultimately influencing which genes are expressed.
It's mind-boggling to think that these tiny chemical tweaks can have such a profound impact on how our genes are expressed and ultimately who we are.
Yeah, it really is a testament to the elegance and complexity of biological systems.
It is.
But it's not just the histones that are involved in this intricate dance.
Oh, right.
Remember those non-histone proteins we mentioned earlier?
Right.
They also play crucial roles in DNA organization and regulation.
Right. You mentioned they're a pretty diverse group.
Yeah.
Can you give me some examples of what they do?
Sure. Some non-histone proteins act as transcription factors,
Okay.
binding to specific DNA sequences and helping to initiate or repress the transcription of nearby genes.
So they're like the conductors of the gene expression orchestra,
The perfect analogy.
deciding which genes get played and when.
Other non-histone proteins play a role in DNA repair, ensuring that any damage to the DNA is quickly fixed.
Okay.
And still others contribute to the structural integrity of chromosomes, making sure that all that precious genetic material is organized and protected.
It sounds like there's a whole army of proteins working behind the scenes
Yeah.
to keep our DNA functioning properly.
It is.
It's truly amazing how all these different players work together to make life possible.
It really is a remarkable symphony of molecular interactions.
Right.
And what's fascinating is that we're still uncovering the intricacies of this symphony.
Mm-hmm.
There's so much we don't yet know about how all these pieces fit together.
That's what makes it all so exciting.
Yeah,
There's always something new to discover.
absolutely.
But, in all this talk about organization and regulation, it makes me wonder, are there times when things go wrong with DNA organization? Could that lead to problems?
That's a very insightful question. And
Yeah.
the answer is absolutely.
Okay.
Disruptions to DNA organization can have profound consequences for a cell and ultimately for an organism.
So what kinds of problems can arise when DNA organization goes awry? Could this be linked to diseases?
That's exactly where things get really interesting and where much of the current research is focused.
Okay.
We're finding that alterations in DNA organization can be linked to a wide range of diseases, including cancer. But before we delve into that, let's take a closer look at some specific examples of how DNA organization can go wrong.
So we've been talking about this incredible dance of DNA organization and how important it is for everything to run smoothly. But, it makes me wonder what happens when things go wrong.
Yeah.
Can problems with DNA organization actually lead to diseases?
That's a crucial question, and it's where a lot of research is focused right now.
Okay.
It turns out that disruptions to DNA organization can have far-reaching consequences.
I can imagine. It's like a well-organized library
Mm.
suddenly having all its bookshelves toppled over. You wouldn't be able to find anything.
That's a great analogy. And just like a disorganized library can make it hard to find the information you need, disruptions in DNA organization can disrupt essential cellular processes.
Right.
For example, if genes that are supposed to be active are accidentally silenced, or if genes that should be turned off are turned on, it can lead to a whole cascade of problems.
So it's not just about the genes themselves, but also about their accessibility and timing. Are there specific examples of how this can lead to diseases?
Absolutely. One area where we're seeing a clear link is cancer.
Oh.
In many types of cancer, we find that the normal patterns of DNA organization are disrupted.
So are you saying that cancer cells have messy DNA?
In a way, yes. For example, certain regions of DNA that are normally tightly packed and inactive might become loosened and activate genes that promote uncontrolled cell growth.
Oh, wow.
A hallmark of cancer, or regions that are normally active and crucial for regulating cell division, might become silenced, allowing cancer cells to escape normal growth controls.
So it's like the control panel for cell growth has gone haywire.
Exactly. And these disruptions in DNA organization can arise from various factors.
Okay.
Mutations in genes that control chromatin remodeling exposure to environmental toxins, even chronic stress.
This is pretty sobering stuff.
Yeah,
But it also highlights how crucial it is to understand DNA organization.
absolutely.
The more we know about how it works, the better equipped we'll be to develop new strategies for diagnosing and treating diseases like cancer.
Absolutely. Understanding how DNA organization influences gene expression is opening up exciting new avenues for disease intervention.
Okay.
For example, researchers are developing drugs that can target specific enzymes involved in chromatin remodeling.
So could these drugs essentially help to restore order to the DNA in cancer cells, making them behave more normally?
That's the idea. It's still early days, but there's a lot of hope that this approach could lead to more effective and targeted cancer therapies.
Right.
And it's not just cancer. Research suggests that disruptions in DNA organization could play a role in a wide range of other diseases, from heart disease to neurological disorders.
Wow, this really underscores the importance of this field.
Yeah.
It feels like we're on the cusp of some major breakthroughs in our understanding of how DNA organization impacts our health.
You're absolutely right, and this is just the tip of the iceberg.
Right.
As we delve deeper into the intricacies of DNA organization, I'm sure we'll uncover even more surprising connections between this microscopic world and our everyday lives.
This has been a truly fascinating deep dive. We've gone from the basics of DNA packing to exploring the dynamic world of chromatin remodeling and its implications for disease.
Yeah.
It's amazing to think that something so tiny can have such a profound impact on who we are and how we function.
It really is a testament to the power and complexity of life at the molecular level.
It is.
And with so much still to discover, who knows what amazing insights await us in the future?
Well, thank you so much for taking us on this incredible journey through the world of DNA organization. It's been an absolute pleasure learning from you.
The pleasure's all mine.
And for our listeners, be sure to check out the show notes for links to some of the research we discussed today. Until next time, keep exploring.